Ladies and gentlemen, we have the honor of Professor Louis Ignarro to deliver the opening plenary lecture for GYSS 2025. Professor Ignarro was the Nobel Prize winner for Physiology or Medicine in 1998. He has devoted his life work to advancing cardiovascular health and is the Professor Emeritus in the Department of Molecular and Medical Pharmacology at UCLA. Professor Ignarro, please.
Thank you very much. It's really a great honor and a pleasure to be here again. I've been here several times, but this is the first time I've attended this GYSS, which proves to be very exciting and a very interesting experience for me. I want to thank the organizers who invited me to come to this lovely place, and the words from the DPM, and also welcome Your Royal Highness. What I'm going to talk to you about is something called nitric oxide. So let me explain first. I'm not going to take the next hour to talk to you about hard science. That would be very boring, not just for you, but especially for me. Believe me. And really what I want to do here is to address the young scientists and try to explain why I asked certain questions, how I answered certain questions, how I was motivated to continue to do my work, and so on. And hopefully by doing that, I can inspire you to do the same beginning in your young careers. So let's talk about nitric oxide. So this is a chemical molecule, a very simple molecule. Before nitric oxide was found to be of biological relevance, it was known to be a pollutant in the Earth's atmosphere, and was first discovered actually by Joseph Priestley back in 1772, and that was 2 years before he famously discovered oxygen. It was Antoine Lavoisier who named the molecule oxygen, you may remember that, but it was Joseph Priestley who discovered it in the first place. So as the title of my talk, when I was going through what I was going to tell the organizers what my title should be, I came up with nitric oxide, the miracle molecule for health and longevity. But then I thought about another title, and that is nitric oxide is not just a gas blowing in the wind. NO is a gaseous molecule like oxygen, carbon dioxide, nitrogen, and so on. And I came up with this title because I was a big fan of somebody called Bob Dylan. Some of you may or may not know him. He released this tune, Blowin' in the Wind, back in the early 60s, 63, I believe. He was from Minnesota. He went to the University of Minnesota. I went to medical school and graduate school at the University of Minnesota. So I got a chance to meet this character, this magnificent singer, Bob Dylan, many, many years ago, before he was famous. I think if I were to try again, it would be impossible to get to meet him. But anyway, if you don't like this title, we can go back to the original one I suggested, and that is the nitric oxide, the miracle molecule for health and longevity. Now, many of you who are not in this area may not know what I mean by this statement, but what I'm hoping is that by the time I'm finished this morning, you will know why I made this statement. So we can begin with that. So this is nitric oxide, NO, simple molecule, one atom of nitrogen, bond to one atom of oxygen, and you can see I'm not going to get into a lot of chemistry here, I promise you, but nitric oxide has a lot of shared electrons here, and that's what happens in order to keep the molecule stable. But NO is a free radical, which means it has one unpaired electron sitting up here. And I mention this because I want to emphasize that this is an unstable free radical. Any molecules that have one unpaired electron are very unstable. They're always looking for other molecules that also have an unpaired electron so that they can pair up to form a stable electron pair, and that stabilizes the molecule. So because NO has this one unpaired electron, it's not very stable. In fact, in the body, it would last for 2 or 3 seconds maximum. But it turns out that that's enough time for NO to do its thing. But it is reactive. It'll react with oxygen species, and it will also react with iron, sulfur, and other elements. So when did this field start? The nitric oxide field started back in 1977. Now, for me, that's not too long ago, but for most of you, it's a long time ago. A pharmacologist friend of mine, Fred Murad, is the one who discovered that nitric oxide can increase the levels of another signaling molecule called cyclic GMP in cells. Now, he made this discovery, and this was all great, but we didn't know what that meant because we didn't know what the importance of cyclic GMP was. There was no known function for cyclic GMP. Completely unknown, complete mystery. And a lot of research had to be done, of course, to figure that out. And many people were kind of upset and that we just didn't know what cyclic GMP did. How could something be present in the cells and we don't understand, why it's there? And then I recalled a statement that Albert Einstein once made, and that is, if we knew what it was that we were doing, it wouldn't be called research, would it? So a lot of research continued after this, and the question was, what was the pharmacological effect of elevated cyclic GMP production by nitric oxide? I'm a pharmacologist. Pharmacologists study the actions of chemical agents on cells and tissues and the interaction between the two. So as a pharmacologist, I began to study the biological effects of nitric oxide added to cells. And in pursuing this, we made the discovery of the first biological effect for nitric oxide in cyclic GMP, namely arterial smooth muscle relaxation. When the arteries relax, they widen. That's also called vasodilation. And so how did we find that NO caused vascular smooth muscle relaxation? Many people ask me, what in the world prompted you to do that experiment in the first place? Did you just pull it out of the sky? I mean, why did you do that experiment? Well, the answer is a very interesting and very truthful story. It's a good thing we smoked cigarettes back then. Let me explain. And I was a smoker back then, but I quit many years ago. We made the accidental finding that cigarette smoke caused profound relaxation of isolated rings or segments of arteries and veins. And let me just show you this cartoon of a schematic of cross-section of an artery. You can see that this is the connective tissue that holds the artery together. This is the smooth muscle layer right here. These are the cells that contract or relax. If those cells contract, it constricts the artery so that very little blood flows through the lumen. And if those muscle cells relax, the artery widens to increase blood flow and also lower the blood pressure. And then I want you to pay attention to this single layer of endothelial cells, which I call the intact endothelium. This is sort of a barrier between the flowing blood and the smooth muscle. And I will get back to that. So the way we do experiments in the lab is this, because I want to explain this experiment to you. We have tissue baths. I mean, this is not complicated, molecular biology. This is simple physiological experiments. We have a tissue bath which is jacketed such that water can flow through to warm the tissue inside in which the ring sits to a physiological temperature, 37 degrees centigrade. And this would be the arterial ring. It's cut like this. The bottom part is mounted to a glass rod inside the tissue chamber. The top part mounts on a rod to a force displacement transducer. We need something to be able to measure the change in length of the artery. In other words, If there's contraction, then the lever comes down. If there's relaxation, this lever goes up. And all this data is recorded on a chart recorder, which is right next to your setup. It's very simple. So we set up about 12 different tissue baths, a dozen tissue baths just like this, with some rabbit coronary artery, the arterial rings, and we're ready to do the experiment. Then my postdoc at the time, Carl Gruder was his name, he used to smoke cigarettes. And he smoked like a chimney, always having a cigarette hanging out of his mouth. In those days, you could smoke in the laboratory. You could eat in the laboratory. And if you closed the door, you can do anything you wanted in the laboratory. I'll stop there. But anyway, he comes in the lab with the cigarette dangling from his mouth, walks up to the tissue baths, and all of a sudden, before our eyes, looking at the chart recorder, the tissues all relaxed. So we thought they died. And he said, "Oh, I'm so sorry, Lou. I shouldn't have been smoking. Let me just put the cigarette out. I'll scrap this whole experiment, come back in about an hour and a half, and we'll be all set to go with some new tissues." And I said, "Carl, wait a second. Let's sit here, let's stand here and wait. Let's see if the tissues recover from this." And sure enough, 8 minutes later, all the tissues recover, and these arterial rings reach their starting level of normal tone. So here we have a reversible relaxation of the smooth muscle, non-toxic. And we didn't understand this. I said, "Carl, go out in the hallway, light a cigarette in the hallway, blow some cigarette smoke into a straw. Come in with the straw and just blow a little bit in the first tissue chamber and see what happens." And sure enough, it relaxes. The other tissues did not. And then he blew a lesser amount of smoke into the second tissue chamber, and we got less relaxation. So we had a concentration-dependent relaxation of the arterial smooth muscle by this tobacco smoke. And so I wondered, what in the world is going on here? So I ran to the library. This was 1979. There's no computers, nothing online, no Google, nothing, just old-fashioned books and journals. I wish that day was still here, believe me. So anyway, I ran into the library, took out some books on components of tobacco smoke. And in the first journal that I picked up and opened up the pages, this is what I read. Cigarette smoke contains about 800 parts per million of nitric oxide gas. That's a lot. If you make a calculation, that's a lot of nitric oxide gas. And the rest is history. So we purchased some purified, authentic nitric oxide. It comes in a gas cylinder, just like oxygen or nitrogen or argon. And we went through all the experiments. So we showed that NO relaxed the smooth muscle and did so through a cyclic GMP mechanism. NO interacts with the smooth muscle, elevates cyclic GMP, and then it's the cyclic GMP that causes the relaxation. So the important point I want to make here is that we were curiosity. We were curious about how tobacco smoke could relax these arteries. And we also found the same thing for veins. We didn't scrap the experiment and say, the heck with that. I mean, it was important to try to understand why that tobacco smoke caused a reversible, non-toxic relaxation of the smooth muscle. And because of that curiosity, we made the discovery that nitric oxide and cyclic GMP are vascular smooth muscle relaxants. Now, at the same time, we were studying nitroglycerin. Why? Nitroglycerin is an explosive. We were not trying to work with explosives in the laboratory, but nitroglycerin, of course, is also used as a drug. Many of you may know that. It's a very potent vasodilator. And it's been known to be a vasodilator since the 1870s. It was discovered in Alfred Nobel's dynamite factories in Stockholm. Nitroglycerin is a volatile, thick liquid. So the fumes get into the air. The workers would come into the factory and breathe these fumes of nitroglycerin and develop tremendous migraine-like headaches. And also, those people who worked in the factory that had angina pectoris, a heart condition, impending heart attacks, were dramatically relieved of their chest and arm pain by working in the factories. And when they went home on the weekend, that pain returned. So to make a long story short, a clinician, a physician in the community, William Murrell, figured all that out. And he actually took nitroglycerin very carefully, tiny amounts of it, a half milligram or less, mixed it with sugar, made compressed tablets, and would give it to the patients with angina in the community. And he found that if you placed it under the tongue, it worked better than if you swallowed it. It's fascinating how these discoveries are made. And so Alfred Nobel is important here because it was his factories. He invented something called dynamite over 150 years ago. An Italian chemist discovered, synthesized nitroglycerin for the first time, Ascanio Sobrero, but he did not want to use it as a commercial explosive because every time he tried to make it, it would explode and killed a number of people or injured them severely. But Alfred Nobel was different. Alfred Nobel, one of his many businesses was a heavy construction business. He built roads, tunneling through mountains and so on and so forth, and he needed a powerful explosive. The best explosive available at that time was plain old gunpowder called black powder, which didn't do much. And so he discovered a way to make the nitroglycerin safe enough so that it could be manufactured and not explode spontaneously. And that's when he developed dynamite and called it dynamite. And because of the dynamite, Alfred Nobel made a tremendous fortune, some of which is distributed each December to Nobel laureates. So thank you, Alfred. Now, the mechanism of action of this explosive as a vasodilator was not known. And we thought, because we were familiar with this area, that it might work through a nitric oxide mechanism. In other words, could nitroglycerin be converted or metabolized to nitric oxide inside the vascular smooth muscle cells? You know, chemistry always gives you the right answer. Take a look at the chemical structure of nitroglycerin. You don't even have to be a chemist to understand, to appreciate this. This is a 3-carbon chain, and it has 3 ONO2 groups. So built into this is NO. It's there. You know, maybe one or more of these ONO2 groups could somehow be converted to NO. It was a plausible, idea. And let me just skip to this. So I just wanted to show you the commercial synthesis of nitroglycerin to make a point. The starting material for the synthesis of this explosive is the very innocuous glycerol or glycerin. Pharmaceutical houses use glycerol as a solvent to make oral preparations to be administered drugs orally. This is a very sweet tasting, thick liquid. But if you take this innocent glycerol and you mix it with concentrated nitric acid plus concentrated sulfuric acid, you wind up with a not so sweet tasting trinitroglycerol, which is the active explosive. So let's look at this chemistry again. So we had the idea that one or more of these ONO2 groups just might be converted to NO. So we did the experiment where we added the nitroglycerin to arterial segments and so on and so forth. We measured products of the reaction, and this is what we found. Sure enough, one of these ONO2 groups was converted to NO. And the second product of the reaction was this dinitro alcohol, propanol. We could buy some of this. We tested it. It was not a vasodilator. So nitric oxide was the active vasodilator principle in the nitroglycerin. And another point I want to make here is that the science we embraced here, I think you could see, was not complex. This is not complex science. Science does not have to be complex to be important. You just need to be able to make observations and then follow up on those observations. And so the conclusion is that nitric oxide was the active vasodilator principle of nitroglycerin. And it was this experiment that prompted me to organize my laboratory, and we now became a nitric oxide laboratory. What else can nitric oxide do besides relax arteries? So being a pharmacologist, we studied the pharmacology of nitric oxide. And we found, as I explained, it's a vascular smooth muscle relaxant, both in vitro, also in animals. In animals, it lowers the blood pressure. It increases blood flow because it's a vasodilator. And in other laboratory animal models, we showed that NO could lower the blood pressure in hypertensive animals. And also, one of my graduate students, Theo Melian, showed that nitric oxide could inhibit blood clotting by inhibiting platelet function. You need platelets for blood clotting. And so NO turns out to inhibit blood clotting and thereby prevent thrombosis. And in another series of experiments, laboratory animal models with atherosclerosis coronary artery disease, we saw that NO had anti-inflammatory properties and could actually prevent the worsening of coronary artery disease and stroke. And so for months and months, I would look at this list of these pharmacological effects of nitric oxide, and I would think, you know, this is amazing. Wouldn't it be something if our bodies actually produced nitric oxide? It would have all these incredible beneficial therapeutic cardiovascular effects. At that time, of course, it was not known to exist in cells. And so the question became then, what is the physiological relevance of all this? I mean, it was all very nice. These are all pharmacological findings. I was able to publish them, but many editors of journals would say, Professor Ignarro, why are you doing this? Why do you continue doing this? What is the physiological relevance? Is this just esoteric work, or is there some kind of physiological relevance? So we addressed the possibility of the production of nitric oxide in mammalian cells. Before 1986, NO was not known to exist in cells. And the reason is it was very reactive, as I said before, with iron, sulfur, oxygen species. It reacted with so many things. It had a biological half-life of less than 3 seconds. So how could such a substance exist in cells, you know, to start with? And then I decided, why don't I just, I mean, my background was in chemical pharmacology, so I have a chemistry background. And I said, you know, I said to myself, forget the chemistry for a second. Just forget it. Put it aside. Look at all the effects nitric oxide produces on cells and tissues. Maybe we should just take a chance and think outside the box and determine if nitric oxide can be produced in cells despite the chemistry. And we did that. I'm not going to go through any of the experimental details, but suffice to say that my lab discovered in 1986 that NO is produced in mammalian cells after all, despite the chemistry. And this was the first demonstration that NO exists in and can be produced by mammalian cells. And they're produced in the arterial endothelial cells. And let me explain that for a minute. So we're back to this schematic again. We looked first at the smooth muscle cells to see if there was any nitric oxide here, because this is the target for nitric oxide. It can relax those cells, causing vasodilation. So why not look there first? Couldn't find any. And then for one reason led to another, and we looked at endothelial cells, and we found that the endothelial cells were the cells that made the nitric oxide. So jump forward, got my thoughts together, came up with this schematic. So here's another cross-section of an artery. So we have nitric oxide is made in the single layer of endothelial cells. So NO is a rapidly diffusible, fat-soluble, tiny molecule. It cannot be stored in organelles or anything. Once it's made, it just diffuses away. Goes through cell membranes, it goes through everything until it reaches its target, and then it self-destructs because it has a short half-life. So, the nitric oxide, I guess the slide didn't come out perfectly, but the nitric oxide, when it is released from the endothelial cells, can interact with the smooth muscle cells to cause relaxation. That's going to widen the artery. In addition, the endothelial cells can actually diffuse, if you can follow this pointer, diffuse into the luminal area by the luminal surface of the endothelial cell. And by doing that, it can prevent platelets from sticking onto the surface and prevent white blood cells from sticking onto the surface. And that could prevent blood clotting and could prevent the development of cholesterol plaques in the artery, as I'll show you momentarily. And a lot of the nitric oxide can diffuse into the blood where it binds to sulfhydryl groups on hemoglobin, stabilizing it, and that can be transported, the NO could be transported downstream, bound to the hemoglobin, reaching sites that need oxygen, that are hypoxic. The NO is released, causing vasodilation and bringing in more blood and therefore more oxygen to those tissues. So in this sort of a video, which I hope works, it'll show the vasodilator effect once NO is released from endothelial cells right here and right on that side. And so when you get that vasodilation, there's an increase in blood flow. And also, by definition, a drop in blood pressure, right? Because you're increasing the diameter of the artery. And as I mentioned before, the nitric oxide could also interfere with the attachment of circulating cells to the surface of the inside of the artery and prevent the deposition of cholesterol plaques and other fatty materials, which you can see in this slide. It takes time, years and years and years. But eventually, if there's a lack of nitric oxide, what happens is that you get deposition of cholesterol plaques, you get a complete blockade of the flow of blood, and when this occurs in the brain, that's called a stroke, and when this occurs in the heart, in the coronary arteries, of course you have myocardial infarction. And so I think there was a lesson learned here, at least in our lab, and that is that sometimes it is prudent to think outside the box. No matter what you think might be happening or what the science seems to indicate, if you have an idea, you should definitely pursue it. And at that time, I remembered a quote made by the famous American essayist and poet Ralph Waldo Emerson, which goes something like this: "Do not go where the path may lead. Go instead where there is no path and leave a trail." And that's what I want to impress upon all of you young scientists. Don't follow everything other people do. Sometimes you have to get somewhere, but then start thinking on your own. That is the best way, in my opinion, to move forward. Now I want to tell you briefly a fascinating story about how evolution resulted in the appearance of nitric oxide. This all started about 3 billion years ago, when all life on Earth was anaerobic, no oxygen molecules at all in the atmosphere. And then for a variety of reasons, carbon dioxide began to accumulate, and very primitive life forms, they were not even unicellular then, these primitive life forms called cyanobacteria reacted to the CO2 and evolved to produce oxygen, analogous to photosynthesis, for example. And then this oxygen accumulated in the atmosphere, leading to what you all know as the Great Oxygenation event. And this allowed for the evolution of aerobic primitive life forms. And as the oxygen production kept increasing, this led to the production of ozone, O3, a reactive oxygen, a molecule in our atmosphere. The low levels of ozone were great. They were very, very protective, protected these organisms against the damaging UV radiation from the sun. But as the ozone kept increasing, the ozone was very toxic to these primitive life forms and began killing them off. And it is told that nearly half of all life on Earth perished from high ozone levels. And then a miracle occurred. And this miracle was nitric oxide. Now, you knew I was going to say that, of course. But the miracle was nitric oxide. Why? Well, these primitive life forms, the cyanobacteria, evolved to produce NO gas to protect them against the rising ozone levels. If you remember your chemistry, NO reacts chemically and very readily with ozone to destroy it, to lower those levels. And so when this happened, the primitive life forms began growing and spreading. Therefore, I think we conclude that NO came to our rescue by allowing organisms to live and evolve further, thereby saving planet Earth from extinction. And we owe this to this beautiful blue-green algae. They were called blue-green algae, but we now know that they are bacteria called cyanobacteria. Blue-green means cyano, or cyano means blue-green, hence the name. So as the cyanobacteria evolved further into unicellular and multicellular organisms, more nitric oxide was produced, serving a variety of biological functions in these organisms. And a great example, what I find fascinating, is the parasite called Rhodnius prolixus, which evolved to use nitric oxide for survival by facilitating blood sucking. They thrived and lived on blood. And here is an example of that. And this parasite would make nitric oxide somewhere in this region. It would bind to a resin. It was then injected into the mammalian skin or surface, whatever. The NO was released. It would cause a local vasodilation. The blood would accumulate and it would suck up the blood. And ultimately, with millions of years of evolution, mammals evolved to form NO synthase, the enzyme protein we know today that is responsible for the formation of nitric oxide from arginine. The NO was used for a whole host of physiological functions to promote health and longevity. And then mammals, believe it or not, evolved to produce NO for penile erection. So here's another role for nitric oxide in maintaining the evolutionary process, I guess. And I'll get back to that. So for these reasons, I just have come to believe that nitric oxide is truly the miracle molecule for health and longevity. I don't know what else to conclude from this molecule. Now, NO is a gas. You can buy it. It's commercially available. It comes in cylinders, so it can be used as a drug given by inhalation. And many of you physicians know that many infants each year are born with a pulmonary vasoconstriction problem. It's called persistent pulmonary hypertension of the newborn. It's an anatomical problem. I'm not going to get into that. But normally when the fetus is developing, of course, the fetus gets its oxygen from the maternal blood, maternal circulation through the placenta. And then when the baby is born, the blood is diverted. Blood flow is diverted to the lungs so it can pick up oxygen from the air by that manner. But in newborns with this condition, that does not occur. So that they're born actually a very blue color. And I can tell you that within seconds or minutes of giving inhaled nitric oxide, these babies turn from a distinct blue to pink color. And this would be what an infant would look like minutes after birth. You can see how purple and blue that color is. And after administering inhaled nitric oxide, from any time between 30 seconds and 2 minutes, the skin just, turns a bright pink. And I was lucky to be brought into the NICU, the Neonatal Intensive Care Unit at Massachusetts General Hospital, by the late Warren Zapol, who discovered this whole procedure. And he showed me this. And I just couldn't believe it. I mean, it literally brought tears to my eyes. The inhaled nitric oxide is also used to treat a variety of cardiopulmonary disorders, which I will not get into, but the one I want to mention here is COVID-19. When COVID first started, some people had a bright idea of determining whether giving safe amounts of inhaled nitric oxide could be useful in treating COVID-19. And it was very safe. In fact, there were clinical trials where inhaled NO was given to pregnant mothers. It completely treated and cured their COVID-19. They went home, they gave birth to perfectly normal babies. And this part of the action of NO in treating COVID-19 was the antimicrobial action, because nitric oxide, among its many other things I told you about, is also antiviral, antibacterial. It inhibits the replication and or kills bacteria, parasites, and viruses. And the coronavirus is the virus in COVID-19, the SARS-CoV-2, and this virus is extremely sensitive to the killing effects of nitric oxide. Patients with COVID-19 die from inflammation and thrombosis in the lungs, primarily a pulmonary disorder. The coronavirus attaches to the lung tissue, destroys the endothelial cells, destroys the vascular endothelium lining all the arteries in the lungs. So you already know what the endothelium does. It makes nitric oxide. You know what nitric oxide does. So what happens when you don't have these endothelial cells to make nitric oxide? You get a great decrease in the production of NO, which results in thrombosis or blood clotting, hypoxemia. You're getting no oxygen there. There's vasoconstriction. You cannot have vasodilation because there's no NO. And so the inhaled nitric oxide replaces the endothelium-derived NO to reduce the inflammation and thrombosis. I mean, it just makes a lot of sense. When this happened, I started advocating that people during the COVID pandemic should breathe a certain way in order to maybe protect themselves against getting COVID-19. And this became very popular, and I learned this procedure because my wife practices yoga. I cannot practice yoga because I cannot move my feet or my legs in those positions without falling over and breaking my neck. But I would watch my wife do these things, and she would make all kinds of noises, and those of you who do yoga know what I'm talking about, and she would be breathing in through her nose. And anyway, a Swedish group back in the early 2000s showed that the nasal mucosa produces large amounts of nitric oxide gas, very easily measurable. The mouth does not. So inhalation through the nose will bring in the nasal NO deep into your lungs. Inhalation through the mouth does not bring in any NO. And so what does this inhaled NO do? Well, it relaxes smooth muscle. So it'll relax the airway, right? The bronchioles cause bronchodilation. That means you can get more air into your lungs. Okay, it causes vasodilation. I've been talking about that for 45 minutes. So that's going to increase blood flow into the lungs to pick up the oxygen in the lungs. And also the inhaled NO will inhibit the replication or growth of the coronavirus. And this is all going to result in greater oxygenation of blood and inhibition of thrombosis. So NO was very successful in treating COVID-19. The reason why so many people have not heard about it is because it's a lot cheaper and easier to give vaccines than it is to put this instrumentation in a clinic or a hospital, which is very complicated to deliver NO. NO is unstable. It is very labile. It could be converted to nitrogen dioxide, which can hurt you. So it takes certain kinds of apparatus to administer the NO. That's why it never became that important. But It was good science, for sure. And so I could tell you that after 40 years of basic and clinical research, I've concluded that nitric oxide is truly the miracle molecule of health and longevity, and I'm happy to debate that point with anyone. Now, I was asked to talk about this, okay? Usually I don't, but I'm going to do it this time. Many people have asked me, Professor, why did you win? I don't want to use win. You don't win prizes. You get awarded prizes. Why were you awarded the Nobel Prize? What did you do that got you to Stockholm? Well, I think there were two studies that paved the way. The reason I'm not sure is because the Nobel Foundation does not reveal details to you. They just give you the prize, you know. It's locked up in some secrets which you can't look up for 50 years. But nitric oxide, one of the reasons was we found that nitric oxide occurs naturally and is produced by vascular endothelial cells. And that discovery was made and published in 1986. And the other one was that we showed that nitric oxide is a neurotransmitter. Of all things, not only is it a neurotransmitter, but it's a neurotransmitter that is responsible for penile erection. And we published that in 1990. And this was the icing on the cake, so to speak. And I'm going to end my presentation with a few minutes of this discussion. We made the discovery that NO is the principal neurotransmitter mediating erectile function. Why was that important? Well, there were no drugs available to treat erectile dysfunction, which affects hundreds of millions of men globally. You know, you don't hear about that disease because it's not really life-threatening. It's life-destroying, but you know, not life-threatening. So we published our observations in 1990 in laboratory animal models, but the human model was published in the New England Journal of Medicine in January of '92. And you know, you all know the New England Journal of Medicine. That's a very prestigious medical journal. This is the only paper I ever published and will publish in this journal. And that was because of the thanks I got from the urologist at UCLA, Jake Rafer, who insisted this be published there. But anyway, so the New England Journal of Medicine publishes profound work, almost in every issue. So the news media, newspapers, magazines, radio, television, they're glued online to wait for each issue of the New England Journal of Medicine so they can jump in and start interviewing people who published exciting and potentially important articles. So I remember that morning I was in the lab. I go in the lab early, about 5 o'clock Pacific time. So New York is like, what is that, 8 o'clock. So about 5:30 in the morning, I get a phone call. It was the first interview. It was from the editor of Hustler magazine. How many of you know Hustler magazine? I think you all know Hustler magazine. But anyway, I was ready to conduct an interview, and then I remembered my mother was still alive, healthy, doing well, and what would she think if her son conducted an interview with Hustler magazine? She was much more worried about what the neighbors would ask her about my conducting an interview. So I respectfully declined and accepted the next interview from the New York Times. I've never had my name in a newspaper for any reason except for this. And it made the left-hand column of the front page of the paper. Not bad for a pharmacologist, I thought. And it went on for another 2 pages. I mean, they really outlined that study. And I thought it was very well written, the article, except that the guy who wrote it, on occasion, instead of writing down nitric oxide, he wrote down nitric acid. Ouch! But otherwise, it was a very nice article. Now, you know, I'm Italian. My parents were born in Italy. And so I can tell you that often Italians think much better, can express themselves much better in pictures than in words. So here was a cartoon that appeared in an Italian newspaper, and it shows a lady in bed with her gentleman. And on this tank is written nitric oxide, but in Italian. Ossido d'azoto means oxide of nitrogen. And you can imagine what my mother thought about this cartoon. And a few months after that, the journal Science decides to declare nitric oxide as molecule of the year. You know, that was okay, but I think they should have done that in 1986 when we discovered that our bodies produce NO. But, you know, we'll take it however we can get it. It's fine. So now, that paper in the New England Journal of Medicine was published in 92. So what happened 6 years later? That's not many. 6 years later, in 98, the Pfizer pharmaceutical company released sildenafil, or Viagra. And the mechanism of action of Viagra, or sildenafil, is to increase the actions of the neurotransmitter nitric oxide in the erectile tissue of the penis. And they invited me to their company in England and showed me, you know, made it very clear to me that if I had not published our work that NO was a neurotransmitter, they wouldn't have developed Viagra, which is fine. I would have preferred a few dollars, you know, of royalty, but I didn't get one nickel of royalty. Rightly so, because, you know, I didn't work for them. So anyway, let's look at the dates more carefully. It was March of 98 that Viagra was marketed. And just a few months later, in October of 98, the Nobel Prize for nitric oxide was announced. And so I'm thinking, was this a coincidence or what? Did Viagra have anything to do with the Nobel Prize for NO? And so I decided to look up the committee members for the prize in physiology or medicine, and I noticed that all of them were men over the age of 60. So I'm going to stop with this slide, but this is the 27th anniversary, actually, for this Nobel Prize in NO. And in my last comment, what I want you to remember from this is not receiving the Nobel Prize from King Carl Gustaf of Sweden, but look at the carpet. Look at what we're standing on. We're standing on NO. NO. You can never get rid of NO. Thank you so much for your attention. Great audience. Thank you, thank you.